Welcome to OncoPharm. I'm your host, John Bazar. I'm a professor of pharmacy practice here at the supporting sponsor of OncoPharm, ETSU's Bill Gatton College of Pharmacy. It is January 15th, 2026, and this is Chapter 2 of a keynote presentation I did about 18 months ago. Chapter 1 was about how doses matter, and that went up sometime last year. And that there really wasn't a lot this week to talk about in OncoPharm world, so I'm going back to this well of information here and slides to talk about how oncology drug use evolves and how that evolution in some ways can be somewhat predictable, although we know it will change. That's what's predictable. How it will change we don't always know, but it does tend to parallel how we learn more about the biology of cancer. So let's start with the cetuximab story. So if you go back now more than 20 years to 2004, cetuximab, a chimeric chimeric monoclonal antibody targeting EGFR, is approved for EGFR-expressing metastatic colorectal cancer cells, which is pretty much all metastatic colorectal cancer cells. Now, it's EGFR-expressing. 5 years later, this approval was narrowed to only those that were KRAS wild-type. Since 2009, the number of patients who are candidates for cetuximab with chemotherapy has narrowed even further to be not just KRAS wild-type, but NRAS wild-type and BRAF wild-type and left-sided as well. Now, there are some data with cetuximab in BRAF mutated colon cancer with BRAF inhibitors, which is another evolution of this. So now there's almost like a split off of this diagram. Now, we got the drug, we gave it to everybody. It didn't work in everybody. In fact, it was probably detrimental in some folks, and it narrowed even further. And this is not uncommon in oncology. We start using a drug. If it looks like it works, we keep using it. Sometimes we're a little bit ahead of the biology. Unfortunately, Martha Stewart was behind on the biology, as we all were, and she actually, I believe, went to prison for some while. She was at least indicted for some insider trading around Erbitux, which is the brand name of cetuximab. Now, this is not the only example of this, where we get a drug, give it to everybody, and then the indication narrows over time. It seems obvious in hindsight, but tamoxifen, a drug that targets the estrogen receptor, we used to give to everyone with metastatic breast cancer, and its use narrowed once we learned it was really just those women who were hormone positive who benefited from tamoxifen. We just went over cetuximab. Meclorethamine is a drug we don't use hardly at all, even though it was our first alkaline agent. Now, in its case, we don't use it because we have learned more about the biology of certain cancers. We don't use it because we have better alkaline agents that do not cause as much secondary leukemias as meclorethamine. And then let's talk about erlotinib. So if we go back more than about 20 years to 2005, this was the pivotal study that got erlotinib FDA approved for non-small cell lung cancer. Now, this was approval in every patient with non-small cell lung cancer. You can see your patients were randomized 2-to-1 to erlotinib or placebo. Median age is 60. About 12% were Asian, which will come into play in future studies we look at. And the makeup of this patient population mirrors what you see in the world. About half of these non-small cell lung cancer patients were adeno. A third were squamous. And most of these folks, they were getting second-line treatment. The other half had probably had platinum-based chemo, and then it had docetaxel, or maybe even pemetrexed, probably docetaxel at this stage. And notice that most of these folks were current smokers. That's kind of what you get in lung cancer. Most of these folks were smokers, and we didn't know the EGFR expression. Mutation status was not something we were looking for. It was just, did these cells have EGFR outside the cell or not? And we see, one small step forward for this patient population, which was all non-small cell lung cancer. We see erlotinib had a median improvement, or an improvement in median overall survival of about 2 months. Hazard ratio of 0.7 seemed pretty good, and this really established erlotinib as the standard of care in the third-line setting for metastatic non-small cell lung cancer. People were excited about it. It was a tablet. It was a pill people could take at home. They didn't have to come to the infusion center, really the beginning of the target area with non-small cell lung cancer. Now, as we're going through this, we knew that not everyone benefited from this. We actually, knew that patients who had a rash early on with erlotinib did better. We knew that some patients, if you're smoking induces CYP1A2. Erlotinib is metabolized by CYP1A2, so some centers were targeting their erlotinib dose up to a rash if they were smoking, getting to higher doses beyond the 150 approved dose. We were really trying to do all that we could for erlotinib in this patient population. Well, then we get IPASS in 2009, and this is the I is Iressa, which is the brand name of gefitinib. Now, this patient population is enriched based on some characteristics of patients, partially because this was done, I believe, in Japan, but partially because folks were sort of identifying, observing, right? An observation, that's the first part of the scientific method, observation, that patients who were female, adenocarcinoma, were Asian, and were never smokers tended to do better with EGFR targeting tyrosine kinase inhibitors than everyone else. So this patient population was enriched with females, 80% female, Asian, adeno, they were all adeno, and most of these folks were never smokers. And these are all advanced, non-resectable, non-curable, non-small cell lung cancer, and they've never been treated. So this is first-line gefitinib or first-line chemo. And what we see if you look at the entire patient population, big picture, there's benefit to gefitinib, although early on we see the chemo folks benefit a little bit more. We see similar curves like this where the curves cross a lot with immune checkpoint inhibitors, also in non-small cell lung cancer. Now this is everybody, 600 and 600 in each arm. If we just look at those folks who are EGFR mutated, which is only maybe a fifth, maybe 20% of this whole patient population, and we see that gefitinib was even much larger magnitude of benefit compared to chemotherapy with carboplatin paclitaxel. We'll see. If we back up one slide, our hazard ratio for the entire population for progression-free survival was 0.74. That drops all the way below 0.5 to 0.48 when you exclude those folks who are EGFR mutated. Now, this is subgroup analysis, so hypothesis generating. Another thing that would help this hypothesis is if we flip the coin. Let's look at those folks who are EGFR wild type. Ooh, you know what we see with gefitinib right there? It's probably the natural history of the disease that's untreated. Gefitinib having no effect in those who are EGFR wild type, and a detrimental effect of gefitinib, an EGFR TKI in folks who are EGFR wild type, again in the first line setting. So to confirm this, everyone knows this, like if you're an oncologist and the patient's EGFR mutated, you're not going to put them on chemo most likely based on this study, but we had to confirm this, and so we get Yordic, which is the European Tarceva versus chemo, Tarceva being the brand name of alatinib. So because we already know the benefit of EGFR TKIs in EGFR mutated patients, not a lot of patients going to be keen on getting on this study. So this is a small study of alatinib versus chemo. Here you can see not everyone's female, but most of them are. They're almost all adeno, and they're not as many never smokers as the IPASS study, but still never smokers, but they all have EGFR mutations. And we know that never smokers are more likely to have EGFR activating mutations than smokers, who are going to have lots and lots of mutations that may also include EGFR mutations, but probably other mutations as well. And so here we see much larger benefit. Here our hazard ratio is 0.037 compared to chemotherapy. And this is, we already kind of knew this for sure, but this really established EGFR tyrosine kinase inhibitor as this first-line treatment compared to chemo for these folks who are EGFR mutated. So if we take a step back, this is a larger picture of a puzzle, which is kind of the framework for this keynote presentation that I did. So, we have these puzzle pieces, and we're trying to put them together, but we don't always know if we have to use that puzzle piece. Like, we don't have the box on the puzzle that tells us what the picture is going to be when we're dealing with all this new information. So we thought rash was a really important piece of the puzzle. That ended up being thrown away. There's a period of time when gefitinib we thought maybe was a worse drug than erlotinib because it got an accelerated approval and then got removed from the market because the phase 3 study wasn't positive. Probably had nothing to do with gefitinib. It had to do with the patient population not being selected for EGFR mutated patients. Then we looked at an enriched patient population besides just everybody. So we had one small step forward for everybody when we only looked at those who are never smokers, really. And that patient population was enriched with EGFR mutated patients. We saw there was a larger step forward, and the biggest benefit we now know today are those folks who are EGFR mutated. So if you were trying to paint or trying to put together the puzzle pieces of how to use EGFR targeting TKIs 20 some years ago, that puzzle looked very different. It was a smaller puzzle, and it wasn't the pieces we were forcing to fit together in a way that really was not accurate. And now we know a little bit more, although hindsight would say it would be hubris to think that we know for sure how these pieces fit together currently, and that will continue to evolve over time. So here you can see a picture. Maybe you know what it is, maybe you don't, but we're going to need more puzzle pieces over time to fill in to know what we're actually looking at here. We're talking about drug use in a specific disease state in cancer. And some of these pieces here in this puzzle may have to be discarded because we get better puzzle pieces, or we may end up thinking, hey, this puzzle piece actually belongs to a different puzzle. This drug does not work well for this disease state. We need to use it for a different disease state. Imatinib's a drug. Puzzle piece fit right in with CML right away. We knew it's a great drug for CML, and it is. Nowadays, the imatinib puzzle piece is used more often for GIST and other disease states probably than it is for CML. Again, our drug use evolves over time as we learn more about the disease and as we get better drugs and as we are able to use our drugs smarter. Well, that's what I have for you this week. Thank you so much for listening. And until I talk to you again, remember, doses matter. Thank you.